I have to say, got it.
Yeah, okay, that should be going now. And I'll just go ahead and introduce you. So everyone, thank you for joining us after a little bit of a break that we had. And we're very happy to have Stephanie Palmer here joining us to talk about her work. So Stephanie did her PhD in theoretical condensed matter physics, I believe, at Oxford. And from there, she moved on to doing postdoc in neuroscience, first at UCSF and then at Princeton. She joined the Chicago faculty in 2012, where she's been working on really interesting problems relating to how the brain accomplishes prediction problems. So if I understand, or if I can give an analogy, how the brain knows to anticipate where a ball is thrown when it senses that it's being thrown, where to move the hands and so on, so you don't get hit in the face.
Exactly.
Okay, good. So with that, Stephanie, go ahead.
Hey, everybody. Thanks for being here. I want to make sure that it's clear that this is a conversation, so please feel free to interrupt me anytime. I'm trained as a physicist, so that's totally cool. What I want to talk to you about today is really, I want to talk to you about the things I'm most excited about in research that we can't quite yet exactly crack with a capital T theory, but I want to tell you how we're approaching it, how we think about problems, anchor it with a few details of a success story. But really, I am happy to pause and dig in and shift gears anytime. So the abstract and title I gave you was this broad thing about how to think about early vision. A better title might have been this, that prediction starts in the eye and has its eye in behavior. And what we're getting at here is that if you want to think about how we sense the world is sculpted by what we're going to do with that information, maybe more so than how an engineer might approach making a biological sensor by thinking about how to efficiently encode everything that's impinging on that sensor. There are some examples of very good coding like that in the history of biophysics theory, but really what that hides is the fact that in those cases, it's always incredibly important to the organism because of its behavioral repertoire, because of its sort of fitness goals to be able to do that kind of processing. So what my group tries to do is to think about neural processing in the rangy, difficult to understand, complex natural environment. So both the natural scene, the visual natural scene statistics that you have, and the natural behavior that you're going to execute. So we'll talk about how we take that apart. So first of all, I want to talk to you about why we focus on prediction. We focus on prediction because if we all ordered neurons from DigiKey tomorrow, we would send them back immediately because they're crappy. And we would say they're crappy because they have intrinsic lags. And if you were trying to build a computer out of the neurons you got, and every single one, A, had just some arbitrary noise that it put in, and B, it had like a couple of milliseconds up to, sort of 10 millisecond lag that it imposed on its input output function, you'd be super annoyed. And why would you be super annoyed? But because you're trying to build something that will have a fluid interaction with the outside world. And we all have the experience of having that fluid interaction with the outside world. We don't feel like we're kind of driving forward in a car with only the rear view mirror and the front windshield blacked out. That would be, the driving fast through the world and looking only through the rearview mirror would be what we did if our brain didn't compensate for those lags. And we know that prediction happens in the brain. And it's important even when the lag seems small. So a 50 millisecond lag for an object that's traveling at 100 kilometers per hour gives you something like more than a meter disparity between what's happening now in your brain and the state of the extrinsic variables in your environment. And this is the requisite tennis player or baseball player interacting with a ball prediction slide that's a talk of all prediction and predictive coding talks. So, you look at this and you see that Serena is not looking at where the ball would have been if she weren't making predictions. She's actually looking not even just at where the ball is, she's looking at where the ball will be. And this is a place where we're as close as we possibly ever will be to professional athletes. Our input visual system is pretty darn good, and there's not a heck of a lot of variation amongst the population, though of course some people's eyes are just faster than others. But really the difference between, say, me as a tennis player and Serena as a tennis player has less to do about my visual prediction in my retina and more about how I control my motor system. So just to say, well, that sounds very arrogant. Obviously, that's everything that it's at. If you're not a good tennis player if you can't control your motor system. Motor system's all where it's at. So you're not just so your eye does this prediction is kind of a baseline start. So what we do in my research program is to try to embed that behavioral goal of making efficient predictions into equations. So here's how we think about it. We think about this as a bottleneck. Here's a cartoon of the external world. This is the path of some object, obviously not a simple tennis ball. It's doing all kinds of jerky things. Here are our spherical cow neurons. So these are the individual neurons, and we're just going to consider their output action potentials or spikes. So these little ticks say where this cell fired as a function of time going across the X direction. And so we consider a population of cells and what they have to say about the stimulus. And we ask how this information gets squeezed down into the representation in the brain. And of course, there's some throwing away. Biological systems are finite, and they have finite resources, finite amounts of ATP to play with. So there is a compression that happens when biological systems sense the external world. And the spin we like to put on this is we like to say, well, there's a compression, but it has to know as much as possible about what's relevant to the organism. And here we're making like a hard guess. We're saying, well, prediction is important. So when this gets squeezed down, it should retain as much as possible about the future. And so this is the bottleneck. By the way, is my mouse showing up when I move it? Good. Okay.
Yeah, it is.
Right. So then what we want to do is put this in some sort of mathematical formula. So we're going to be solving this Lagrangian, where we're going to say we're going to minimize the information we have about the past. That's the throwing away the part, subject to constraint that we keep beta amount about the future. And that's the keeping what we are guessing is relevant. And that's how we've kind of put our notion of what the behavioral goal of the organism is into an equation form. And for the purposes of this talk, you can think about information. Maybe some of you know a ton about information theory and it's all familiar to you. This is just the mutual information between the stimulus in the past and the neural response now. And this is the mutual information between the neural response now and the stimulus in the future. If it's not familiar to you, you can think about it as a generalized form of correlation that goes to all orders of possible correlation between two variables. Okay, so we want to solve that. And we want to look to the retina.
Sorry, Stephanie, can I interrupt quickly?
Oh, absolutely.
Can you go back to that slide? Can you just repeat? So is this is reading like off of one neuron, or like what is this example supposed to be? The data.
Oh, this example is saying there are many neurons in a recording. This is time. And we're going to look along here and ask, say, at this instant in time, what is the neural response word? What did the brain say in a small time window? What does that tell us about what happened in the past and what will happen in the future? So of course we're going to have to.
The wave is some behavior thing.
What's that? Oh, sorry. This is the external. This is the trajectory of some external object in the visual scene. So if this were the tennis ball, it'd be a crazy tennis ball. But this is exactly the stimulus that we project onto the retina, which is a moving object that's moving in 1D. Here, that 1D is up and down as a function of time.
So if it's just a stimulus,
yeah,
then where does prediction come in?
Right. So the prediction comes in how the brain represents what the stimulus will be doing in the future.
Oh, I see.
So time is here.
The stimulus from in the past. Okay.
Yeah, and of course, this whole problem is sort of ceilinged by the stimulus correlations between this blue box in the past and the blue box in the future. There is no fortune telling. We're not looking at fortune telling for what it's worth. We're looking at, given the correlations of your outside world, how much do you capture in your neural response?
Okay, thank you.
Absolutely. So we're going to look in the retina. It's interesting to look in the retina because it's actually part of your central brain that's sitting in your eye. So unlike the sensors in your fingertips, it's not just your peripheral nervous system, though it kind of feels like it might be because your eyeball is kind of out on this stalk and separated from the rest of the brain. That's nice if you're a theorist because you don't have to think at all about feedback. Whereas if you're in the, sort of meaty part of your brain, everything's connected to everything to first approximation, and you have to be careful. Here, the biology of it has given us this nice place to play because it is part of your central brain, so it has some computational complexity, and it's sort of isolated. So if you can put it in a dish, you get a complete picture of what the brain sees, and there's some neat processing going. So there's evidence that I won't get into that says that part of that prediction about where I'll say moving ball will be in the future starts in your eyeball. And so if you take a slice through your eye, your retina, the tissue at the back of your eye there, it's a complex piece of neural tissue. This is the input. These are rods and cones. So your rods and cones. And there is a lot of guts of the retina that do complex processing. So just by sort of biological parsimony, you can squint at this and say, this isn't a pixel-like camera where the pixels are the photoreceptors. Something's going on here. And these output axons are the cable that comes out of the back of your eye. And that's what creates your blind spot for what it's worth, because these things have to punch back through the retina. So we're trying to understand what's being coded in this cable, this collection of axons from the ganglion cells. And the ganglion cells tile space at the back of your eye. These ganglion cells have incredibly diverse functional response properties. This is a image from a paper from Thomas Euler's group. You're not supposed to be able to read it or take it. You're just supposed to be like, wow, there's a lot of different stuff here. So even if you're a lumper versus a splitter, you find that there are many, many categories of response properties to dynamic inputs to the retina. So these cells are pretty diverse, and there's a lot of complexity to the retinal circuitry. So like I said, the retinal cells tile space. So if we smushed the retina down on a glass slide and we were looking just at those ganglion cells, this is what their dendritic arbors would look like. This is a cartoon from Vaslow's review, which is a great place to start learning about the retina if you're interested in it. And this is a particular subtype of ganglion cells, and they have dendritic fields which pool from some part of space, and that pooling kind of tiles all of visual space. So if you know what the brain sees in an area from these neurons, you kind of know what the visual world was doing in that corresponding part of the external world. Our textbook pictures of these cells is that they have a kind of circular, symmetric, quote unquote, receptive field. And here I'm displaying it as an off-type receptive field. This cell is excited by light offsets. So if we were doing an experiment on these cells, we might flash a bar, and this is literally as fast as you would flash it in an experiment. And hopefully you can see that black bar flash as long as there's no zoom issue. And you can see what that lag is. So here's the firing rate of one particular neuron. So it goes up from 0 spikes per second to 400 spikes per second, a really sharp response. Here's the time. Oh, yep. Question.
I had a question about the ganglion cells. So
Yeah.
You said that it's some information processing mechanism, like it's some processing the firing that's there in the retina. So do you lose information, or you condense it, or like what's exactly happening if you have some, like what is the ganglion cells actually doing there?
They're doing a lot, and that's really a fascinating question. It turns out that the ganglion cells, by the time the light goes from the photoreceptor layer to that's the input layer to the ganglion cell layer, there's a huge number of different nonlinear processing steps that happen. And what's interesting about that a diagram that I flashed up from Thomas Euler's group is that different subtypes of ganglion cells have different nonlinearities. So is stuff thrown away? definitely information is thrown away by some groups of ganglion cells, just in terms of like how they sum over space and things like that. And there are so many that you could argue that you kind of put some of it back together. But in terms of like the light that hits the photoreceptors and what you output, there's a massive compression. And what we'd like to understand is what that trade-off is about what gets thrown away and what gets kept. It's also important to say that when that processing happens, you don't get to reevaluate the output from the retina. You don't get to kind of think it, you don't get to undo it. So you can't think your way around any kind of maybe illusion that your retina is spitting out at you. So it's really important that the retina provides you with all the information you need to do behavior. Great question.
Thank you.
Okay, so here's our lag. Here's one ganglion cell. Here's the time where we made that dark flash, which that was a dark light offset preferring cell. And this is the response. And you can see here, the time between 0 and this peak is roughly 60 milliseconds. There's a lag. And if you do an experiment where you flash the bar again, but you're able to record from, say, a whole population of cells, then you can look at what their response profile looks like. Now you can look at it as a function of space on the retina. If we collapse along this Y direction, just onto the Z direction relative to the center of the bar, and then we can look at that kind of retinal image as a function of time since we flashed the bar. And that's what this is. So this is position in microns on the retina, which again corresponds to some, position in space that's, some degrees of visual angle out in the real world. And here's the firing rate of the population as a function of space. 0 is the position where we flash the bar, and these different colors are 44 milliseconds after we flash the bar up to 62. And 62 was the lag of that one cell. And you can see that there's a buildup of activity that comes in because the cells have different lags and different, they have some biological variation, but that the peak response happens, at a pretty hefty lag away from the time 0 where we flashed it. So in the range of 44, where maybe some cells are firing that's just barely detectable that there was a bar there, to this kind of neural image of the bar, which is, a Gaussian kind of blurred image of the rectangular bar we flashed. So the question is, this is the thing that the brain has to compensate for. And what do we know exactly about those nonlinearities that happen in the retina? So what if we do something different? What if instead of flashing the bar, we drag it across the retina? Oh, another question.
Sorry, this might be naive, but how do you measure the firing rate in these cases? And I'm curious about that because the frequency of the firing rate is on the same order of time scale as the different time steps where you're measuring the activity.
No, you actually are able to sample the activity at 30 kilohertz. So we can really do screaming fast. This is all extracellular. I'll have a picture of the recording array, but basically what you've got is a glass slide with embedded electrodes in it. And that kind of extracellular recording lets you go screaming fast in terms of how you're sampling. This is there's no voltage die or anything that would make this kind of impossible.
Oh, okay. Thanks.
Yeah, absolutely. And that's another advantage of playing around in the retina. It's to some extent it's a flattenable tissue, and you haven't chopped up all the connections when you pull it out of the eye. So if you can carefully put it down on a glass slide in which you've embedded electrodes, you have a linear array of neurons and a linear array of like a 2D array of neurons and a 2D array of electrodes that you can push against each other, and then you can get what you need. It's part of the reason that theorists gravitate towards the retina. It's a pretty beautiful prep where you can get good population responses. Okay, so like I said, we're gonna drag this bar across, and we're gonna look again at what this one cell does. And now time 0 is going to be where the bar was when we drag it, where the bar is when we drag it across, relative to the point where we flashed it before. So we're dragging this bar across now the retina. We're giving a movie to the retina. And 0 is the time where that sweeping bar reaches the position where we flashed it before. So you can see that when you move the bar across, and that was centered on the cell's receptive field, this 0 point, you can see that you get a lot of activity way before the bar reaches the center of the cell's receptive field. And that is an element that will really help you make predictions. There's all this activity up to hundreds of milliseconds before that bar arrives at that position, where you have already the retina responding in a way that could help you do prediction. So if we do the same experiment, but we record again from many neurons, and we're going to do the same kind of projection down onto this x-axis of position on the retina, we see that what we had before when we flashed and we looked at a 62 millisecond lag is that we had this neural image of the retina. If we look at the same kind of, we take the neural response when the bar reaches that position where it was flashed, we see that the neural image is already kind of pitched towards the direction that the bar is moving. So this is the motion anticipation nonlinearity that you see in retinal ganglion cell processing. And what that, and it was discovered by Marcus Meister and Michael Berry when he was a postdoc in the Meister lab. And what that tells you is that when you have a moving stimulus, there is a front of neural activity on the retina that doesn't just lag behind it, it actually starts to move out in front of that moving object. Now that's for a bar that's sweeping in a completely deterministic, constant speed, constant direction motion. It turns out that there are all kinds of other nonlinearities that have a hint of prediction that have to do with motion. And so when this is just sort of a note for students and postdocs who want to take this down and dig in more. There is the reversal response, which was identified by the Berry lab and carried out by Greg Schwartz, it was your very own Greg Schwartz when he was a grad student in the Berry lab. And that shows that there's a volley of coordinated spikes in the retina when a stimulus reverses course. And there's also things like omitted stimulus response, where if you have a regular stimulus and it has a gap, there are a bunch of cells in the retina that'll respond to the gap or respond after the end of a train, but at the appropriate lag, as if they've kind of become entrained to the rhythmic input you put in. And object motion sensitivity from Bence Olvetsky when he was in the Meister lab. So there's a ton of nonlinear processing. It all has kind of various hints of prediction, but what our group decided to do was to try to take this apart kind of holistically. So what we're going to do, move beyond this kind of motion, and we're going to, or that kind of motion, and make a more complex stimulus to show to the retina. And what this has is a mixture of predictable and non-predictable parts. So the bar just moving and sweeping at a constant speed is wholly predictable. So the extent to which you know about where the bar was tells you all you need to know about where the bar will be. So it's hard to really know how much is bona fide prediction going on in the retina versus, okay, I've adapted to that current motion, and I can just prospect for it as much as I want because it's all deterministic. The reversal is like a completely unpredictable thing. It's going along and then all of a sudden it changes course. So that sort of says, how do I code for something that's surprising in the retina? What we wanted to mix here was both predictable and non-predictable. And we want to ask, when the retinal ganglion cells do their compression, do they preferentially throw out the non-predictable part? So this bar motion, a lot of you are physicists. This is familiar. This is the velocity of the bar. This is just your F equals ma equation. This is V is velocity, X is position. And then there are some parameters. So this is white noise with some diffusion constant D. If we ignore that for a second, the rest of the equation is just a damped harmonic oscillator, where there's some omega naught spring constant tethering it to the center of the screen, and there's some tau describing, together these describe the damping. So what this noise does is give us something like a Brownian motion with this restoring force. So we have kind of a stochastically driven damped harmonic oscillator. And it's a kind of motion that's rich enough. It has both predictable and non-predictable components, because those are predictable parts, that's the non-predictable parts. This is where the bar lives as a prior. But you can tell here that motion is where it's at, and we can solve a lot of pieces of this kind of motion analytically that makes it really attractive for us to look at as a stimulus to present to the retina. So it's simple, but complicated. So what we want to say is, here's an equation that we're going to try to solve analytically, and we're going to ask how the retina does solving it too. So this is our notion of optimal prediction. If you minimize this function, you will have an optimal amount of information about the future. So some amount here, given how much you compressed about the past. So if you have lots and lots and lots of information about the past, you can kind of saturate how much you know about the future. And it saturates at some level because there's noise in the system. So with some measurement of the past, you top out at how much you can predict about the future because there's noise. You can't get into this gray region. That's the no fortune telling rule. And this bound curve is parameterized by beta. How much you want to know about the future tells you how much you can compress the past. And if you go beyond that, you can't have that much. And you can think about it either way too. You could say, if I keep this much information about the past, how much could I possibly know about the future? This curve tells you that. So then the question is, where does the retina sit? And the retina can sit anywhere it wants, as long as it's not in the gray forbidden region. And what we find is that the retina sits close to the bound. So how do we do this? Again, this question came up already. We anesthetize a live animal. We dissect out its eye really carefully, and we place it into a dish where there's a glass slide with electrodes embedded in it. So if we zoom in on that, these are the electrode pads, these are the ganglion cells, these are their axons. And we've got a good match between the density of electrodes and the density of the ganglion cells. So we can see what the brain sees in that region. And we do this in salamanders and in collaboration with Olivier Mar in Paris. Some of my students have gone there to visit Olivier and worked with him. They also record from rodent retina, so mouse and rats. And this is just one of the more modern 252 electrode arrays that we put the retina on at 252 because the four corners are ground pads. Okay, so we want to measure optimal prediction in the brain. We're going to put this movie onto the photoreceptor layer. We're going to record retinal response. The retina is, for small groups of cells, compresses this much. So we're going to zoom in on that part of the bound curve. And what we find is that the retina response really hugs the bound. So for 1, 2, 3, up to 7 cells, we see pretty good sticking to that bound. And the error bars here are both the error in our measurement of those information quantities. It's a hard thing to do, just practically data speaking, and also the difference in different populations of neurons we see. We can't really tell if this peeling off is, leaving the bound as we get to larger populations, or if we're just running out of data. So that's a TBD thing. And we see the same thing happen in the rat retina. So this is salamander and this is rat. That if we plot the information we have about the future versus the information we have about the past, the neurons really seem to be close to the optimal bound that we calculate. So it's pretty cool. It says we made a guess about what might be an efficient way to do prediction in the retina, and it looks like the retina does that. Now, you could start to—
Stephanie,
oh,
I
yes.
have a question.
Yes.
This is really fascinating, by the way. I'm really enjoying
cool.
this
Thanks.
talk. Here in this case, I'm just wondering, how do you know how much information the rat has?
Yeah, so that's the nitty gritty of how do we compute these things. So here's a little diagram. What we do is we take a trajectory of the bar stimulus in the past. It's just a 1D thing, so we can sample that pretty well. It's Markovian, so we really only need to sample 2 position steps, and then that's like a sampleable thing. And but we ask about the mutual information between, say, those 2 positions of the bar and the full binary response of the retina. So that's a 2 to the n vector, and that's why n doesn't get very big, because we've got to fill out a matrix to characterize the mutual information, that's the joint distribution of stimulus and response. And so the response is 2 to the n, and the stimulus is however many different pixel positions there were squared, because we're doing 2 of them. So that becomes a pretty rangy big vector. That's why I said this requires you to have a tight relationship with your experimental colleagues. And I actually did some of these experiments at Princeton, because you have to dedicate a whole experiment to just sampling this space well enough. But that's how we do it. It's kind of a direct, straight up, direct mutual information calculation.
Okay.
And we do it the same way for salamander and rat. And if you want to push to larger n, you can be tricksy and employ some techniques from like Ilya Nemenman's group about doing unbiased estimators of info, so you don't get like hung up on sampling errors, and you're confident you're doing the kosher thing.
Okay, got it. Okay,
Yeah.
I guess in this case, it's just that you really do know what the in the actual experiment what like a neuron is supposed to be firing if it actually like has the predictive information of the input. That's the part that I'm trying to understand.
Oh,
like,
that is a great question. Sorry, I was just giving you the like details of how we make the calculation. I think you're asking.
Yeah, I'm asking about the actual data points.
Yeah, you're asking a more nuanced question, which is pretty sweet. So the data points do come from that mutual information calculation, but there's no what they should be doing in terms of a binary collapse. So this bound curve is calculated on a continuous variable, and there isn't a constraint that I've put in on that bound to be a projection onto an n-dimensional binary vector. So that's a pretty cool result in and of itself, that there's already some sort of like data processing inequality here that says we're going to go from this continuous space down into this binary code that we could have been away from the bound just by virtue of that. Mm-hmm. And so it seems like for what it's worth, that kind of sort of binary response is set up so that it does make up for that, which is pretty cool. Oh, hey, Louisa, my gosh, nice to see you.
Yeah, hello, nice to meet you too.
Yeah, Milo, I hope that was satisfactory.
Yeah, thank you.
Yeah,
great.
So I have a question about, like, if you increase the noise in the stimulus, you will in principle decrease the threshold in this information plane, right? So somehow it doesn't matter how much you know about the past, you will know nothing about the future because you have a lot of noise in the stimulus, right?
Yeah, that bound curve, as you add more noise, is going to take this just totally down. And that's a very good, well, I don't want to anticipate your question. Go ahead.
Oh, so I was just wondering if you can somehow vary that amount of noise in order to know something about like the capacity of the retina itself. Like if you have a totally deterministic stimulus, how much the retina can actually
Right. So as you vary, so you're imagining that we're going to dial up or down the diffusion constant in that equation of motion. And so that's gonna change the bound curve. So one way that we've done that is a little bit of a hack. It's not quite directly changing the amount of noise, but of course the other way that this curve collapses down is if you look further and further into the future. So this is for a lag of about 16 milliseconds. You can trace this out at prospecting 100 milliseconds in the future, and so forth. And eventually, past about 200 milliseconds, you don't have any information left. So what we did in this 2015 PNAS paper was actually trace that down all the way till it falls apart. And we find that there are some groups of cells that just trace the bound all the way till it ends. So that's fun. But the next thing we did, which is closer to what you're asking, is to play with the stimulus statistics itself and ask, we didn't exactly ask about sort of capacity. We were more interested in what is I'll give you the like how we did science answer, like the real, there's a story I can dress around it, but here's the real answer. If we keep finding that the retina saturates the bound, we should worry. because maybe it's doing that just because there's a non-trivial consequence of a very trivial feature of neural encoding that we haven't quite grasped. Like if every time we look to see if it's optimal, you might start to get a little queasy. So what we decided to do next was to dial up and down the damping coefficient in that equation motion and just see if we basically feel like we should be able to push the retina to its limit where it stops doing optimal prediction somewhere. It should cut out. Because again, otherwise you worry, like you have to be able to push something till it breaks to understand that you've really understood how it's working is the idea. So what we did is we created different stimuli that in this instance were constrained to have the same kind of static distribution of positions and velocity. So these, it's a little hard to see because of the jerkiness of zoom, but if you gave the position and velocity distribution of these two movies, they would be identical, but the dynamics are where it's at. And this one is underdamped and this one is critically damped, but otherwise they're statically identical. And we asked, if we play with these parts of the equation of motion, Luisa, you're asking about this one, what do we get? So when we do that, we find that for this kind of underdamped motion, the neurons look optimal, and those are the data I showed you before. And these same neurons look nowhere near the bound for the critically damped. Which to our eyes looks a little bit noisier because we might say, okay, it fell off the edge of the, kind of speed sensitivity of these neurons. What's fun is that these neurons have maybe even more information about the past, about this stimulus than about this one. But they can, sort of bridge that gap into prediction, probably because what Jared has figured out so far is that it's because of their particular kind of velocity and position tuning. So the rat retina seems to be set up to optimally predict motion that falls in this category, but not here. And that makes us feel better because if we paste on top of this a textbook linear nonlinear Poisson model of what these neurons do, like just a linear filter with an output nonlinearity, nothing saturates the bound. So it's nice to see that sometimes they look like our old vanilla textbook neurons, but for certain stimuli, they seem to have like evolved a way of really doing a good job of prediction. So this is that result that I'm hinting at, that if you make a linear nonlinear Poisson model and you give it every advantage in the world, you even let it encode more about the past than you saw in your neurons because you let them not be redundant. But so you really max out that they match exactly what the single neurons did. They are not saturating the bound, and indeed they look like they're saturating a bound, but with 100 millisecond delay. So the difference between the plain vanilla textbook model and what the data tell us is exciting, and the fact that not everything is optimal is reassuring. So those are some of the things we're digging into more. And I want to just take a pause and ask about questions here, because I'm going to switch gears and tell you about something that's much, much harder to analyze. We can't do anything really analytically, but I think it's a fun result. Yeah, Lisa.
Yeah, so I have a question about if you have think on maybe doing that in outer layers of the retina. So I know that their signal is continuous, so it's really hard to do that, but maybe I will guess that the information will also saturate, like for instance, in photoreceptor layers, we will not expect that neurons to predict. So somehow we will get better in different layers of the retina.
So this is, yeah, no, this is wonderful. So, well, maybe I don't need to black this out. So the question is, if we took this step by step, could we watch this evolve, and could we then get at some circuit mechanisms and, nitty gritty synaptic, what exactly does that? And I think that's wonderful. We're trying to push there in the mesoscopic way, just by looking at, what kind of elements you have to add to your LNP model to get it to do this. But the other thing we're looking at is we're trying to get some of our colleagues who work on bipolar cells to measure the same thing. So that'll be fun to know. I don't have an answer yet, but watch this space.
I was wondering about these movies. They're like hard to watch, right?
Yeah, they're not fun.
So is that like is that, I almost would think that our brain or maybe even our retina cells are like trained to not be able to interpret them at all.
Oh no, so they're hard to watch. Part of that is Zoom. Sorry.
Oh, really?
There it's better. It's better when it's not when it's played at the proper sample rate. It's also part of the magnification where this would be a big object in your visual scene moving. You have to think of it more like fluttering leaves or something like that. It's more salient. It's also in these critters that aren't our eyeballs.
Right.
So they have different speed tuning curves. So the hard to watch thing is important in just how you tune up the stimulus to start. So when I was a postdoc at Princeton, we spent more than a year just playing around with different statistics to try to get the cells to respond. And what we found when we had the sweet spot was when the information processing was of the same order of magnitude as when we showed natural scenes that were relevant, that were at a scale relevant to the salamander, to these neurons. So that was how we kind of benchmarked whether or not it was hard to watch, I guess, for the salamander retina in the dish.
Okay.
But point taken, it's important to kind of gut check some of these things.
All right. Okay.
But all those velocities, sort of snug into the speed tuning of these cells. We're not like completely pushing outside the bounds of what's viewable to those retina.
Makes sense.
Yeah.
Thanks.
Stephanie, I've got another question. So in the previous slide that you showed that the rat eye is better tuned, or maybe it's 2 slides ago. Yeah, the rat eye is better tuned for some instances of some parameter space of your stochastic process than others. Is there evidence that this information is actually actuated by an organism? So like if you were to show a scene to a bar to a mouse and track its eye movement, would you then predict that it's better tracking better in one scenario, not the other?
So it's a really good question. So what have we done to do this? We don't have the tightest answer yet, but you're exactly right. The first thing we did is we started collaborating with Jason McLean, and he's got awake behaving mice that are on a treadmill. They're head fixed, and he's doing 2-photon imaging of their visual cortex. So it's not quite retina, but what he can do is he can track their eye movements while they watch movies. And they're running along, so that makes them a little more activated and interested in things. So we have eye movements of mice watching movies. So then you can put those eye movements on top of the movie or things. I mean, you can just ask whether or not this is sort of behaviorally relevant. One thing that's tough is that, of course, when the mouse moves through the real natural world, it's moving as well as the objects in its scene moving. So we can't, from these data alone, disambiguate whether or not this is like the mouse or rat moving its eye in a complex natural scene as it's running away from the hawk that's trying to eat it, versus it's sitting, rodents very seldom sit and stare, whereas salamanders do that a lot. Salamanders are sit and wait predators, ambush predators. So in a way, the experiments we do are more suited to them. So the way that we've tried to crack this is one exactly as you say, with recording eye movements while the alive, awake animal watches these movies. The second is to analyze the statistics of natural scenes and see if we can figure out whether or not there's an ecological niche argument for who predicts what best. That's a little harder, obviously, to sort out.
Super interesting.
Awesome, thanks. Okay, so of course, you want to be able to push this a lot farther. So I think we're getting at those questions already. It's this is neat, but is it behaviorally relevant for the animal? So can you find an instance where prediction operates in a complex natural environment while the animal's doing a complex natural behavior, hopefully one that's mission critical. So this is I was hoping Madhav would be here. This is, maybe Madhav is here in the background. This is a little, well, so this is a fly. This is a fly that's flying. This is data from the Dickinson lab, and it's going to receive a very terrifying stimulus. So one of our colleagues in Comp Bio, who's really scary because he's good at everything. This is Ilya Nemenman. And Madhav and I could have a chat about that, have a laugh. But anyway, so I didn't want to put anybody from Northwestern up here. But so Ilya scared this poor fly, and you can see that it does this banked and counterbanked turn and flies away from the threatening stimulus. And flies do this fast on the wing escape behavior. So let's just watch it again because it's pretty fun. So this is 400 times slowed down. This is Drosophila going screaming fast. So the fact that the Dickinson lab can even record this is amazing. There's a slight lag after the threat comes in, and then there's the banked, and then you'll see the counterbank turned. So there's a pitch and roll axis here, and you can see the fly's head moving. It's elaborate. So the thing that's cool about this is that this is a behavior that is mission critical. So you have to escape from a predator. That predator will eat you. And if you just kind of throw yourself away from the predator, like flies do when they're walking on a surface, they just kind of launch themselves in an arbitrary direction. That'll work when you're walking, because you can just sort of throw yourself at the world, and you're close to the surface. So then you can just kind of try to hide. If you're in the sky and you throw yourself around, you'll just fall very predictably towards the earth. That's not good. You will get eaten. And we know that flight is aerodynamically unstable in these guys. And also that if they just do a banked turn, they will slide out of the sky. Early inventors of the airplane had to figure out how to make the little flaps on the wings go. And so this has to be exquisitely controlled. So when you're going to do a fast banked and counterbanked turn, you really have to do it well. But here's the problem for the fly. This all happens in 40 milliseconds. And there is a 20 millisecond lag before the halteres, the little gyroscopes on the fly's back. Instead of a second set of wings, flies have these little gyroscopes that help them sense their position in space. That haltere has a completely wrong gain setting because it's used to just doing regular flight. I'm not, escaping. Escaping takes an order of magnitude different angular velocity. So there's this lag. The halteres are set up to give you feedback, but they won't have anything to tell you for a while. And visual feedback is going to take even longer. So if the loops in the fly brain are faster, but they're not there, its behavior is screaming fast too. So based on this and some new data from Brad Dickerson in Michael Dickinson's lab, and now he has his own lab at Chapel Hill, there's a new connection in the fly brain that seems to go from the vertical sensing system of the fly down to the neck motor center to directly impinge on the place in the wing steering muscles that could help you modify your haltere gain. So our hypothesis, and this is work of Siwei Wang, a postdoc in our group, was that the visual threat comes in that lets the animal compute a feed-forward visual prediction that could input here and help you bridge this gap. So it's going to do this exquisite ego rotation control, and it's going to get help from feed-forward visual prediction. So that little gap before it starts moving gives it enough time to create the signal that says, okay, I know where I'm going to be when I throw myself out of the way here. And so what Siwei did was she simulated the fly visual system traveling through a natural scene. This is supposed to look like a natural scene. These are images from the Van Hatteren database of natural scenes. So there are trees here. It's a 3D world. This is just showing one hemisphere. And that visual scene is going to be then modified by the real escape flight behaviors that were measured by the Dickinson lab. So this is a plug for open data in science. Dickinson lab published all of those super fast escape behavior trajectories for lots and lots and lots of flies. They published the whole darn data set. And Siwei was then able to implement them in silico and move a whole visual scene with natural scene statistics around as the fly was like flying through that fictively. The other thing is that by collaborating with Axel Borst and Idan Segev, we know exactly how to like implement the vertical sensing system of the fly. So we need to filter the visual input both by the fly's natural behavior and by natural scene statistics. But then we actually know what all the components are. You can make a pretty good in silico fly vertical sensing system because Axel can tell you exactly what all the values on all these different electronic components are. So you can build this in your computer. Seaway can. And then there are 10 VS neurons in all flies, and all of these are blowflies. But the 3 in the center are a triplet that have that connection, that suggestive connection down to the neck motor center that can impinge on the steering control system in the fly. So what Seaway did is she did this whole thing. She said, okay, here's the natural behavior. Here's the circuit that's implementing it. First of all, she has to compute what the bound curve is, but you can see there's nothing Gaussian or nice or well-behaved here. There's no analytics to do. So she threw all of her computer science training at it. That's what her PhD is in. And so she could use all the most modern variational info bottleneck and other techniques to solve the bound problem. Then she could ask, how does this in silico network compare to that? And then she could just start ripping up the circuit. So what she did was compute the bound. So this is, again, the information about the past, information about the future. We'd like to be on the bound. If you look at the output of all of the vertical sensing systems, so all 10 cells together, you hit the bound pretty darn close. If you just look at triplets and all the different triplets, they also sit near the bound, though they're a little reduced because you just, sort of don't have as much output. And there's one little piece of the circuit where if you wipe out that connection, you hamper your ability to do prediction. So this is the gap junctions between dendrites in there. So she even figured out like what the element is that makes this all hang together and work. Now, the fantasy experiment would be put some sort of optogenetic label in those gap junctions, because if you just knock out the gap junctions writ large in the fly, it doesn't survive. If you knock them out in the vertical sensing system, they don't fly. So you kind of want to turn them off on the fly, so to speak. That's, an experiment for another day, but she could do it in silico and say, that takes you far from the bound. So to summarize, what we work on is on making better stimuli and better measurements. We try to show that optimal prediction happens. Stuff I didn't have to tell you about was about biologically plausible readouts of this information. And of course, we're pushing towards natural behavior. We're pushing across species. Let me see. There's some other stuff I'm gonna skip. There's some more, how can we push this theory further? Some really nice work from Vedant Sachdeva in the group, but that's in PLOS CB last year. You can go check it if you'd like. And then there's a bunch of stuff I haven't had time to tell you. One of the joys, one shouldn't just give you like the drinking from the fire hose version of what we do. But one of the joys of being a theorist in biology is that you can work on all sorts of different stuff. So we get to work on camouflage and cuttlefish, and we get to work on butterfly, mate selection, evolution, and neural computation. And we get to do some like nitty gritty, let's dig in and talk about the kind of statistical physics approach to neural coding. So with that, I want to thank everybody in my group who makes this possible. Here's Sue, who we just talked about towards the end. Here's Jared, who thinks a lot about natural scenes and their statistics. And our 2 terriers who keep us all entertained with their natural motion behaviors. And people give us money. And so questions.
Amazing. Thank you so much. Any questions? Go ahead and raise your hand or just sort of unmute.
Sorry, Karna, are we in the, are we switching over to the ask me anything section?
No, not quite yet. So just before that.
Not quite yet, okay.
Yeah, right. So this is, if anybody has questions pertaining to the research content of this talk. But it appears that I think we can just switch over to the ask me anything part of the talk. Okay, so I'm going to stop the recording.